Here we go. Lesson 1, DNA and proteins. Now, I'll be studying a new series, year 12 biology, and hopefully I'll do a series for each subject I'm taking. So it's now year 12. A lot of changes, right? Since the last time I made a video, many things have happened, but let's focus on biology at this moment. You have to go through the learning intentions to know what you are doing. So 1.1.1 DNA stores and transmits genetic information, which functions in the same way in all living things. It functions the same way. Okay. Now, what this means is all living things, all have one thing in common. We all have DNA. So every living organism has DNA. And they function the same way in all living things. How do they function? Well, we're going to get into that later. That leads us to 1.1.2. DNA is a helical double-stranded molecule. That means it kind of looks like if you imagine you have two strings. You pull the two strings straight and you connect them on both ends. And then you just kind of twist them. So that would be a double helical structure. Let's go to DNA, the molecular life. And these are just some fun facts to get you interested in year 12 biology because it's a very hard course and you gotta be very determined to finish the course at a high standard, at a high level. Inside our body, there are trillions of cells. Inside the human body, there are trillions of cells. And each cell has 46 human chromosomes. And so in your body, there aren't just one type of cell. There are different types of cells, obviously. There are skin cells for skin. There are liver cells, your cheek cells inside your cheeks. And you can actually get some of your cheek cell by rubbing um, a stick, like a cotton stick, whatever you call that, against your cheek. And you can place that under a microscope and observe your cheek cells. They're alive. And each cell, no matter what cell they all have 46 human chromosomes. This means they all contain the same information. And they all have 2 meters of DNA. They all have 3 billion DNA subunits. Now, what this basically means is the bases. The bases are A, T, C, G in DNA. And there are approximately 30,000 genes coded for proteins that perform most life functions. So it's pretty complicated inside a thing that's very small that can be only seen through a microscope. Okay, the next thing we learn in year 12 is nucleotide. The nucleotide is one of these little things. They make up your DNA. They make up the double helical structure of your DNA. And a nucleotide is made up of three things. You have the sugar in the middle, which is represented by a pentagon, and you have the base, which is represented by a rectangle usually, and then you have the circle, which is phosphate. You have sugar, phosphate, and base, and the bases could be ATs or CG. And A stands for adenine, G for guanine, and they are double ring, but we don't need to get into that right now. And there are cytosine and thymine, which are smaller pyrimidenes, and they're single ring. Next, we go to prokaryotic and eukaryotic cells. In prokaryotes, DNA is bound to proteins in linear chromosomes, which are found in the nucleus. Important thing to know is linear chromosomes are the X-shaped chromosomes you see in mitosis. So they're the X-shaped one. And they're really, really condensed. So condensed that, for fun fact, they're the most condensed information storage ever we know so far. So inside a chromosome, your chromosome is actually the most condensed information storage. Then more condensed than supercomputers, more condensed than a hard drive. It's super condensed and stores the blueprints to make up your body, make up the blueprints to make you who you are. Okay. 1.1.4. DNA is unbound and circular in the cytosol of prokaryotes and in the mitochondria and the chloroplast of eukaryotes. Remember, it's a misconception to think that in eukaryotes there are only chromosomes. There are only X-shaped chromosomes, but that's not true because in our mitochondria and in a plant's chloroplasts, there are actually unbound and circular DNA, and they are not called chromosomes despite some people call them chromosomes, which is a weird thing. But yeah, there are also circular and unbound chromosomes in mitochondria and chloroplasts. Now, this is something that we have learned about for years. Prokaryotic are relatively small, simple. They range from 1 nano to 2 nanometers, which is quite a small scale if you think about it, for life to exist. They possess rigid cell wall which prevents rupture of the cell, and they don't have membrane-bound organelles. They're absent in prokaryotic cell. Genetic material is not enclosed in nuclear membrane. This means their DNA is just floating around in their cytosol, and the genetic material consists of single or endless double helical molecule of DNA. This means they're relatively simpler. And eukaryotic, they are complex larger cells. They range from 10 to 100 nanometers. There is no rigid cell wall in vertebrates, but in plants, cell wall is present. So in humans and in animals, there are no rigid cell wall. But you see in plants, there are cell walls. Membrane-bound organelles are present in eukaryotic cell. We have mitochondria, we have RAF-ER, those all are organelles that perform complicated tasks, and they are amazing. And they are not present in prokaryotic cells. Genetic material is enclosed in a well-defined nuclear membrane. This means our DNA is in a nucleus. And since prokaryotic cells don't have a nucleus, their DNA just floats around in the cell, in the water of the cell, which we call cytosol. There are many chromosomes, form of linear, open-ended, double helical. I just spelled something wrong here, molecules of DNA. There are many different types of chromosomes. And they code for different things, basically. Okay, location and arrangement of DNA. Prokaryotes, DNA is unwound floating in the cytosol. Definition for cytosol, the liquid inside cells around the organelles. Complex mix of substances dissolved in water. So simply put, cytosol is the liquid that's in the cell without the organelles that's in it. And cytoplasm is the liquid that's in the cell with the organelles in it. Okay. Eukaryotes, DNA is wrapped around histone proteins, which then wound up to form linear chromosomes. Imagine DNA as a string, and you wrap the string around a bobbin. And this bobbin is the histone proteins. They are basically the bobbin for the DNA. And so DNA wraps around this protein bobbin, which makes it very, very condensed. That's how you store strings and sewing strings. That's how you also store DNA likewise. And then you have all these histones. They then wrap up to become more condensed, which may seem like a mess, but they just kind of wrap up even more to become more condensed, and they then make up chromosomes that's in our cell. So chromosome breaking down is actually just wrapped up things, just wrapped up DNA, which are really condensed. Okay, DNA replication. 1.1.5. Replication of DNA allows for genetic information to be inherited. And 1.1.6. Base pairing rules. I just spelled something wrong again. That's bad. DNA replication are universal. Okay, key properties of DNA allow allows it to copy itself exactly, so to pass on information to offspring. Yeah, I'm missing some words inside the sentence, but doesn't matter now. I'm too late into the video, and I'll probably correct it later. No, I'll probably not. Okay, polynucleotide composition. Okay, we learned about what nucleotide was. Nucleotide was you have sugar in the middle, you have base as represented by squares or rectangles, and you have phosphate as circle. Polynucleotides is just made up of many repeating nucleotides joined together. So imagine you have a nucleotide right here, and it's laid out horizontally. They join together horizontal. They join together like pancake, right? Imagine you have a pancake flat, and that's your nucleotide. When you stack one on top of another, that's your polynucleotide. And your DNA, just imagine, is a chain of pancakes all stacked really tall together. But that's not possible because, um, you know, gravity. And these can be broken apart and new ones added. So your pancake chain can be broken apart. You can just take it apart and add a new pancake in there and put it back together. So that's a pretty cool property of polynucleotide, in my opinion. And the second property that of DNA that allows it to copy itself exactly and to pass on information to offspring is nucleotide pairing. If you have one polynucleotide strand of DNA, you will make the other side the same every time. This means you know how the base pairs how A always pairs with AT and C always pairs with G. Likewise, if you have just one strand of DNA, if you have A, T, C, G, you know the other side will be, what did I say? T, A, G, C. Yeah, it will be the opposite. It will be the opposite um, pairing partner of the valence strand. So that's pretty straightforward because we know that before from year 9, whatever. And the third property is weak bonds between strands. Okay, so a double helical structure, you have two strands of DNA. And between these two strands, what's holding them together is a hydrogen bond. What's so good about hydrogen bond is they're so easy to break down and reform, which is good for DNA replication. If you look at them one at a time, sorry, if you look at a hydrogen bond one at a time, they're actually pretty weak. You can break them apart very easily. But if you look them as a whole, so you have many weak hydrogen bonds in your DNA that together hold together the DNA. That force altogether is going to be very strong. So it's like if you try to rip apart the whole thing, like ripping apart a phone book, it would be impossible. But if you do it once at a time, like if you just take a piece of paper and rip that apart, that would be super easy. And so when the DNA is getting ripped apart, the enzyme that's breaking apart the hydrogen bond, does something that's similar to ripping a page at a time. It's like a zip, right? A zip doesn't just rip apart the whole thing in 1 second. The zip goes from one end to another end, unzipping, each segment at a time. So that's easy. But if you try to pull a zipped up jacket in half, that's going to be near impossible because all those forces joined together is going to be very strong. Okay. DNA replication fork. DNA replication is a semi-conservative replication. This means the new DNA strands are made up of one strand from the original and one strand of newly made. Results of semi-conservative replication: there are two new double strands that are identical, and each new DNA strand has half of the original DNA. Okay, so this will be the original template, and here you see the double helical structure is ripped apart, and now we have two separate strands, and the blue represents the strands from the original template. Now we have the red ones added in some point here, and they are the new ones that are added in. Where do the red ones come from, you may wonder? Well, the red ones came from free-floating nucleotides that are just in the cytoplasm. So imagine this will be in the cytoplasm, and in the cytoplasm there are just free-floating nucleotides, not polynucleotides, but small nucleotides that are just floating around there. And they are ready to be added to a new DNA. Why are these two identical, you may ask? Because when they separate, this strand will be the exact opposite of that strand. And because the base pairing rule and because a new chain is added in, this new chain will now be identical to the blue chain on the other side. And this blue chain will be identical to that one on the other side. So that's why you have newly synthesized. That's why you have identical, newly synthesized DNA, and that's why it looks the same. And now onto my favorite part: the enzymes used for DNA replication, the helicase. The helicase is the zip that I was talking about. Helicase unzips DNA by breaking hydrogen bonds. So helicase does the job of this. And you have DNA polymerase, which adds free nucleotides to expose DNA strands. Polymerase does the job of adding the red strand in. Now, if we see here, we can realize one thing that so the helicase is working its way before the polymerase. Of course, you have to unzip the thing. So it's working from here to this direction. Pretty simple. It's unzipping the strand. And now you have DNA polymerase, which is adding the strand. And because a DNA is anti-parallel, that means they are parallel, but they go in two different directions. One DNA polymerase has the easy job of just adding missing nucleotides from that point on. But the other one has to go in the opposite direction, and it's a more complicated process, but we don't need to know that at this moment. Fun facts. And now we're near the end. Almost all cells have DNA except red blood cells. Now remember how I said before that all cells have 46 chromosomes. They all have DNA. Well, that was a generalization that is false. We know that red blood cells don't have DNA, and that's the only exception. Secondly, it stores all information from the manufacturing and functioning of every cell, every tissue, an organ, the whole human. So DNA is basically the blueprint for being a human. Well, I can't be bothered to read out the facts. You can read them in your own time. And at the end,